So the question that we were asking is, you have rare copy number variations. Most of them probably have no effect on clinically important endpoints. But what if there are some rare mutations that are still important for the patient? How can we detect those rare variants? Here you see an example. It's not about copy number states, but it, I think, better visualizes the problem that we have. So these are ovarian cancer patients from the TCGA cohort. And this is chromosome 7, so each row here is a patient, each column is a position on chromosome 7, and each dot is a mutation that the respective patient has. And then this blue line is P53, and as you can see, and as you probably know, most ovarian cancer patients have a mutation in P53. Importantly, not all of them. There are a few exceptions. So we know what P53 is doing, but what about all these other mutations? So most of them are probably just passengers. They probably have no clinically relevant impact on the survival of the patient, but some of them may. And it's very difficult to tell which mutations are really important for the patient for survival and which ones are not. So the idea of the study was we wanted to address the following questions. Which mutations are potentially relevant for the patient? And again, here we only focus on copy number changes. How important are they? What are the clinical endpoints? And what are the molecular mechanisms? What are the downstream effects of these mutations? In this presentation, I can't address all of these points, but you will see that we work towards them. This is the outline of the study. It's a combination of different modeling efforts. First of all, we wanted to predict what is the transcriptional impact of copy number changes. So when you amplify a gene, you often increase the expression of that gene, and how will that expression change then have indirect effects on other genes? Or if you delete a gene, you get a reduction, and what is that reduction going to cause downstream? So in order to answer this question, we trained a transcriptional regulatory network. There have been many of them, probably thousands, I don't know how many. But in this case, importantly, we used a quite consistent set of expression data, copy number data from the CCLE consortium from the Broad Institute. So here we used cell line data for which we have copy number and expression state. And then we used Lasso to learn a model which allows us to predict downstream expression changes. And then we validated this with independent data from the TCGA cohort and with other perturbation data that I'm not going to talk about. And then on the other hand, so now we can predict what is going to happen if you have a copy number change, what's going to happen at the expression side, but we also want to know how is that important on the clinical endpoints. So which genes have to change so that you see a change in this case in lifespan or survival. So here we were using TCGA data for which we have survival data, and then we trained a random forest model, that's this thing here, to predict survival given expression. And this modeling effort allowed us to identify marker genes, or signature genes as we call them, that are predictive for the lifespan or the survival of the patients. And now we can put these two things together, because now we can look at a specific copy number change. With this model, we can ask what are the expression changes and how are the marker genes changing their expression if you have a copy number change over here. So then we put this together in scoring that we call an impact scoring, which allows us to essentially predict the average or summed impact of the copy number changes on the survival of a specific patient. So this is really going towards personalized medicine here. Okay, how does it work? I'm skipping almost everything regarding the validation. I just want to show you this one figure here. Because the transcription network was trained on cell line data, and now we're using it to make predictions in vivo tumors, which is obviously very different. So it's an important question, how transferable is this model? And it turns out to be actually quite predictive. So here you see the correlation. This is the correlation between our prediction and the observed expression in the TCGA cohort. And then this is just the histogram for all the genes. And what you see here is an average across 13 different tumor types from TCGA. And then we did an additional filtering here. So you see this is very, very positive. So most genes have a positive correlation between prediction and observation. That's nice. But then we did an additional filtering by removing interactions in the lasso that are not significant. And then this even further increases the predictive power of the model. So you see that this bump here is appearing with even more positive correlations between the predictions and the observed expression values. We did this for individual tumor types, of course, and of course it's not the same for different tumors, but it was always like this, so there were no big differences. Now this is the result, or an example of the results that we got. This is for glioblastomas. Here you have the human genome, and then when you have a bar that goes up, that's an amplification. If a bar goes down, it's a deletion. And if you have a long bar, it's a high impact on survival. If you have a short bar, it's a low impact on survival. And the color encodes the frequency of the mutation. So dark means frequent, and light, like pinkish, is rare. And if you work on glioblastomas, then you will immediately recognize this amplification on chromosome 7, which is quite frequent and well known. And then there's this deletion on chromosome 10, which is also well known for glioblastomas. Now you could, for instance, use that to investigate more these deletions and amplifications and find out what are the genes that are actually important for the tumor phenotype here. We did that, that's in the paper, but here I want to focus more on the rare mutations. So these less known pinkish mutations, some of which were actually quite important according to our scoring. So first of all, how many copy number changes or how many copy number changed genes do you have to look at in order to fully explain the change in survival for each patient? So basically, how many mutations are impacting on survival? That's the question here. So here in these graphs, you see the cumulative impact that we have if we consider several copy number change genes together. And then here, each line is one patient. So we start with the specific mutation of a patient that has the highest impact, then we add the second one, the third, and so on. And then so you're looking at the cumulative impact up to 1,000 genes. And you see that it always plateaus at some point. So this plateau here is basically the maximum impact on survival that we can predict with the CNAs alone. And then, of course, on top of that, you have small mutations, you have treatment, and all the other clinical factors that are relevant for survival. The important take-home message here is that the number of CNAs that you have to look at is very different for the patients, and it can be up to almost 100 genes that, as we predict, still have an impact on survival. So you may have 5 or 8 genes that are initially important for the neoplastic transformation, but then there are additional genes that also impact on survival through other means. Now this is addressing this question about the rare versus frequent mutations, how important they are. Basically, this graph is showing the average impact of mutations as a function of the frequency, how often they occur in the population. This is glioblastoma, ovarian, and this is lung cancer, different colors. And then on this axis, you see the frequency. So this is 20% of the patients in a given cohort have that mutation. And you see that the more frequent mutations have, on average, higher impacts. And this is exactly what you expect. This is deletions, this is amplifications, and of course you expect that the more frequent mutations will be more relevant for the tumor phenotype. That's what you see here. But this is just the average. What if you look at individual genes? This is the same axis here, but now we're plotting each gene as a single dot, and we have here the impact scores. Now important is that these are actually distributions here. And here at the lower end with the rare mutations, of course, you have a huge number of genes that are rarely mutated. But then you still have some outliers here that have very high impacts. And these impacts can be as large as the impacts of the frequent mutations. This is for glioblastomas, just the deletions, and then we can look at amplifications. We see a similar picture. Yeah. And then you look at the other tumor types, and again, you see the same. There are some rare mutations that are as frequent as they are as important as the frequent mutations. So rare really means in this case 1 or 2 patients, right? This is 1 patient, this is two patients, and so on. So now this is a bit puzzling, right? Because here we have mutations. If you, let's say we take this slice here. So here we have mutations that have about the same impact according to our model, but they have very different frequencies. So how does that happen? Because if you take a tumor evolutionary standpoint, you would argue that mutations that are beneficial for the tumor should also be frequent. That's it's not a perfect argument because tumor evolution happens inside the patient, and so we're looking at the competition of cells inside the patient, whereas our study is across patients. But of course, you would still assume that these more important mutations should also be more frequent. So what's going on here? We first considered that might be there are different functions involved, so that you have frequent mutations that are important for the initial neoplastic transformation, and then you have mutations that kick in later, which are maybe important for drug resistance or metastasis, something like that. I'm not showing you the data, but it wasn't really very convincing. So we didn't see a clear functional separation between the rare and the frequent mutations. So what we thought is maybe it's not the function of the genes, but it's the genomic structure that is important here. The genome structure is impacting on which regions are more likely to mutate or less likely to mutate. And that's what we're trying to show here. So here on the left-hand side, you see lung cancer. And then here you have the distance of a mutation to some genomic feature. In this case, we're looking at the distance to tumor suppressor genes. And you can see here that amplifications, frequent amplifications, tend to be further away from tumor suppressors than rare amplifications. And this makes total sense because, of course, the tumor doesn't want to amplify tumor suppressor. So if you have an amplification of, for instance, an oncogene that's close to the tumor suppressor, the breakpoint of the amplification has to be right between the tumor suppressor and the oncogene. And this is, of course, very unlikely to happen if these two genes are close. If the genes are far apart, that's more likely to happen. That's exactly what you're seeing here, this difference. Here's a similar picture for glioblastomas and fragile sites. So these are sites in the genome that are more likely to break. And again, you see that frequent, in this case, amplifications are closer to fragile sites than rare amplifications. So it's also the physics of the chromatin that impacts the frequency of these mutations. Here are two other examples. Again, for lung cancer, here we're looking at the distance to oncogenes, and then you see the opposite as what you were seeing with the tumor suppressors. So now the deletions have to be far away from the oncogenes because the tumor doesn't want to delete the oncogene. And here you have the distance to essential genes, and you see that, of course, you cannot delete essential genes, and therefore deletions have to be further away from essential genes. So conclusion is we think that it's more the physical and, if you like, geometric structure of the genome that determines the frequency of these mutations rather than the function of the genes. So that brings me to the conclusions. First, it was surprising for us that it was possible to do this modeling, to create a model in cell lines and then apply to very different tumor tissues. It works surprisingly well for us. That's basically the biological conclusion from that. There are a lot of regulatory relationships that are actually conserved across tissues. We think that's underlying this, this observation. Rare variants can have large impacts. That's what we're showing here. Most of them don't, but there are these few exceptions which might be important for the individual patient. We think that the frequency of the copy number alterations is really driven by the genome structure. And what I didn't say is that these observations were also very tissue-specific, which is supporting recent publications that the mutational processes themselves are very tissue-dependent. So it depends on the number of cell cycles you've gone through, but it also depends on chromatin structure, the physical makeup of the cells, et cetera, et cetera. All these factors impact on where you get mutations or not. So here are the acknowledgements. Most important is Michael Seifert. He did almost everything here. I didn't do anything. And he got some help from Betty, but he's a former postdoc of my group, and he did really a fantastic job here with this project. I can't stay until the coffee break, unfortunately, so if there are questions that you have beyond what we can address now, you can just send me an email to this address. So thank you very much. Okay, then just send me an email if you have one.